Okay, thank you very much for joining our seminar series today. I'm really happy to introduce Dr. Zhong from the Washington University in St. Louis. Actually, I am studying plant hormone and then she's studying epigenetics. So we don't have much course in terms of the research, but although there is more and more in epigenetics and more in hormonal regulations. But anyways, I met her in Hawaii 2 months ago, 3 months ago, yeah. Hawaii ASPB meeting. So during the break time at the beach, not in the and then I was there. Then my husband talked to me like whispering, "Do you know you're the only one who has a life jacket?" Among the adults. And I was feel so bad, and I was there. And then some woman is walking toward me with another professor in agronomy, and wearing their life jacket. And she's one. Actually, I have very positive interaction. So, and then I searched her and then for research, and then she's working on epigenetics and really cool. And also recently there's ethylene plant hormone regulation. It's also do some important crosstalk with the epigenetic regulation. So I think it's very good addition to our department seminar. So I'm gonna briefly introduce her background. So she did her BS and master in China, Wuhan University, and did a PhD in Ohio State University. And then she moved to California at UCLA for her postdoc in Stephen Jacobson's lab. And then during that time, she is a superstar, published many good publications, including Cell. And then she got a position in Wisconsin medicine in 2013 and stayed for there until 2020. And she found a new home in Washington University in 22. So she's now a full professor there. And her lab is interested in understanding how the cellular machinery can sense internal and external stress or cues, and how the chromatin dynamics is regulating in terms of plant adaptations. So the specific focus is, I think when I read is chromatin, I guess a little bit of a, chromatin dynamics in relation to epigenetics, in how the plant can adapt to environmental stresses. So I think with that, I think I can smile. Yes, welcome.
Well, thanks, Xiaoming, for I hope that I pronounced it right. For the nice introduction and also the recap of our Hawaii trip. It's actually really great experience that meet the familiar people on the beach, right? So it's a pretty good experience. And so it's also, I would also take this opportunity to really kind of thanks for the invitation. It has been so wonderful to meet so many great kind of colleagues and also meet wonderful students and postdocs during lunch. And so it's really, really kind of like remind me myself when I was in kind of like a graduate kind of like a school, how I'm eager to read speakers and engage them and then get all the information from them. And really that you guys remind me 10 years of like the past. So you see me 10 years of you now. So yeah, so as you kind of all know that our lab are really centralized to understanding epigenetic regulation and primarily focusing on the developmental and environmental plasticity. And so for those of you who are not at the hardcore of epigenetics, and I'm always thinking about, what is the difference between genetics and epigenetics, right? And then, so I'm not going to talk to you, Damon, so let's keep silence for now. But I actually thinking about, I also watched this kind of very fancy movie called Superhero, right? So how many of you guys have ever watched this movie? Wow, great. So what do you see here, right? If you're thinking about the parents, the superhero gene is genetics. And then all the three kids has this superpower ability, right? But yet, they have all different superpower. So the older sister that has this very, very kind of invisible superpower, and the boy Dash actually run very, very fast in the movie, faster than a car. And then the little boy Jack actually have a so-called like a superpower. At that time, the movie set is yet to be discovered in the next movie. So I'm really looking forward to the next movie of the superpower movie. What exactly the Jack actually has a superpower? So this is actually using this cartoon really to kind of like just like as an analogy to what is the genetics and what is epigenetics. And basically genetics is like the genes that you have from parents, and the epigenetic is really talk about the phenotype. So even you have the same parent, your sibling, identical twins, you could have differential kind of phenotype at the end of the day. So, this is really thinking about when I started after getting a PhD, and I'm looking for what I'm going to be doing as a postdoc and eventually becoming my own lab. And so that whole field since like early 2000. And so this field has been really driving and then demonstrate the importance role of epigenetic regulation in almost all aspects, ranging from basic science, medical, and also the agriculture. And showing here is just really some type of example in diseases, and this is the identical twin, and then the aging, which is now a very, very important topic, right? And also the diet and the stem cell. And so what we are very interested in the environment, right? We always talking about this changing climate environment, how epigenetic has this plasticity really to kind of respond in the ever-changing environment, and how can we utilizing this basic knowledge to transform the plants, engineer the plants to be more climate resilient plants. And so my lab is really kind of centralizing. The key question is linking the epigenome violations. We talk about the constant, the term is violations. Everything is viable, right? So how can we use linking the violation to developmental and environmental adaptation? And so specifically, we're very interested in what kind of signal or particular signal stress for development that triggering the epigenetic modification. And we know that from all our genomic studies, genetic studies, that certain epigenetic modification is not random. They always have particular patterns in the genome, in the different region of the genome, and in certain developmental stage. And so we're very interested in what is the machinery to ensure the right deposition of epigenetic modification, like right time, right place. And what's wrong if you have mistake happens, right? So you have misdeposition of the epigenetic marker in certain place, wrong time. What happens? And so we are very interested in looking for the plant traits and also stress response and epigenetic inheritance. And so our long-term goal is to translating the basic knowledge of epigenetic regulation and doing like so-called machine learning, phenotypic prediction, and epigenome engineering. At the end of the day, we really want to kind of find a way to engineer plants to becoming more climate resilient kind of adaptation. So for my lab, that we are focusing on two key epigenetic pathways. And the first one is the DNA methylation, and also histone modification is a modification on the histone tail, in particular H3, give you example. And so like a listing here is over the last 5 years that our study has been really centralizing to understanding some type of basic mechanism of DNA methylation, and also how the stress, kind of we call it environmental regulation of the DNA methylation. And recently we start to be very interested in the population and the evolution of DNA methylation. And we realized that we talk about DNA methylation importance, but not every single species has DNA methylation. So what is evolutionary kind of significance of having DNA methylation in plants and in humans, right? And so we're also very interested in the crosstalk between the DNA methylation pathway and the histone modification pathway, particularly focusing on this DNA methylation crosstalk with H3K9, which are another silencing pathway in the plants. And recently, we're also finding that there's a connection of the DNA methylation with some type of H3K9 or the acetylation mark. And of course, we are also interested in the barrier aspect of the histone modification. And show here an example is the K9 methylation and also a group of HDAC, which is a histone deacetylase that remove and deacetylase chromatin, and that involved in various areas of the plant immunity, heat stress, and the ribosomal switch. And so we also have interested in the K27 trimethylation pathway, which involved in PRC. And so I'm going to share today very briefly a story that how we identify this called epigenetic switch by regulating the key process of developmental transition. So, speaking to this epigenetic variation, and we always talk about the changing is expected, right? So we're always thinking that changing is expected, and we are very interested in looking for the epigenome variations. And so this is inspired by this called 1001 epigenome project. They're looking for all the DNA methylation from a global collection of Arabidopsis accession. And showing here is that kind of map from Europe, and looking at the individual kind of like blue and red dots independent methylation status of accession isolating of that given location. And so the blue is lower DNA methylation, and then the red is a higher DNA methylation. And you can see that there's a greater violation of DNA methylation patterns, even isolating from different regions in the Europe. And so indicating that environmental has a kind of influences on shaping epigenome. And so when we also talk about at the individual level, and we have a serious discussion today, right? So even within the same Columbia that we actually reference kind of Arabidopsis growing almost in a lot of Arabidopsis lab. And so we recently reanalyzing a lot of data, and we're realizing that actually within the same Columbia from same different lab, they actually also have DNA methylation violation. And showing AB here is just leaf and individual is a methylome, they are whole genome methylome. And also even tissue-specific methylation variations. For example, showing here is just like we talk about flower, embryos, leaves, and things like that. And then also we call it the cell type specific DNA methylation. Methylones looking for using this PC1, PC2. So it's all kind of, this is basically that the idea is that methylation variation occurs at the individual tissue and cell type, and also in the populational level. And so the key question that we are trying to understanding is why methylation always vary, right? And so what is the function and what is the mechanism? And so we are really trying to understanding this key question in both individual and then populational level. So before I start to tell you the results, I would like to take this opportunity to introduce these kind of key methylation enzymes. So in the mammals, that the DNMT1 is maintained, and then the DNMT3A is de novo accession methylation. And whereas in plants are more complicated, besides this major CG methylation, which is MAP1, plants has this non-CG methylation, which is maintained by 73 for the CHG, and 72 and the DRM2 for the CHH. And then the DRM2 is the enzyme that doing the de novo methylation in all three sequences. And so that becoming very interesting when we talk about mammalian. I don't know whether we have mammalian people in the audience, that if you look at the structural and then the functional and the domain wise, that the mammalian version of DRM and then the plant version are almost identical. And so here showing the structural kind of like a overlay between the mammalian version, which is the gray in the and then the plant is in the light blue, and so they are almost identical. And yet we all know that the version in the case of the DNM3 in the mammal is predominantly in the CG methylation, and so very little or very minimum kind of activity on this non-CG. And whereas look at the DRM2, that is called CHH. If you look at the H, that basically anything but not C, and then you make the mass, it's actually non-combination. And so this suggesting that somehow DRM2 has this called the substrate diversity pattern. And so we are really trying to understand why these two enzymes act at a structural, molecular, domain-wise are so kind of similar, and yet they have such a distinct functional kind of in the plants versus in the animals. And so to really address this molecular basis, we are really collaborating closely with our collaborator, Zhong and Jianfang from the UC Riverside. And this work has been really driven by my talented graduate student, Sarah, who is now postdoc at the Hutchinson Cancer Institute, and then postdoc Zhong, who is already established his own lab. And now, like over the 2 years, the lab is bigger than mine. So it's like a really great kind of like a team. We are very fortunate to work together. And so our first approach is to, let's see, like really then look whether there's using the structure approach to really solve the crystal structure of the DRM2 with the CTT DNA. And so, you know, to make a long story short, because this is always part of the publication, is what is the most exciting part of this structural approach is we finding the deformation. So I want to pause here. What is deformation in here, right? So if you look at this, is a B form, which is a gray, is a regular DNA in a kind of double helix structure, right? But if you look at the green one, which is the CTD DNA that bound by our protein. And what you see, major group and a minor group and major group, you see the deformed. Both major and the minor are deformed in a way that, you know, so which is meaning that suggesting is very interesting that our enzyme actually deform the DNA. So maybe this is how the way to increase diversity. And so this is how and then we next want to know, is this really true, right? And so we took the other advantage of our structure because we can identify which residues are involved in DNA binding. And so we finding this key residues called the 397. And so we are trying to using a lot of biochemical approaches. Let's see, we training all possible amino acids of this C and see what happened to the enzymatic activity. And so interesting, there's two groups that came out. When you look at this is our wild type. And so if you changing the C to all those like a small uncharged amino acids, they are maintaining this kind of like wild type activity. However, if you changing this C to this Q and R, and they are all large and charged amino acids, they are somehow reduce or abolish the enzymatic activity. And so that's interesting that like really tells us something that they are favor a group of stuff rather than individually, right? And so when then we talk the more phylogenetic approach and looking at in the plant kind of like dynamic species and looking for what in the conversation of this C397 position, and we finding that all the plants have all similar like valine, cytosine, and alanine. They are all small uncharged amino acids. And so this is really speaking, you know, this is kind of important. And so next, a logical question is, okay, if this is important, what if we mutating them to R, right? And so we did the same thing that now basically this is a DRM2 and this is a CR mutation. And when you look at that, we find indeed changing the preferences now becoming CTG, which is shown in the blue here, in vitro. And so what about in vivo? Same thing that we're using the in vivo, basically using this genome-wide DNA methylation analysis, and then in complementing the DDC, which is loss of the CG and loss of the non-CG. And we find that in the case of the green line, which is the CR mutation, we see a greater activity of CHG methylation even compared to the wild type. And so suggesting this single amino acid change actually changing the subject preferences from the CHH becoming CHG. And so this is quite interesting to us. And so we are thinking about that maybe the reason that why the mammalian version DNMT2 kind of is so specific to CG is still using this traditional one enzyme, one substrate mode, which is called the nucleotide recognition. And yet DRM2 is maybe because of the deformation, so it's like a shape. Shape is actually DNA shape recognition that mediating this diverse substrate. And so after this, that people thinking like, oh, that's great. Then this is very specific to the method transfer. So what about other things? Does it happen to me also? So actually, yes, of people who are working on transcription factors. A lot of time you are thinking about that, my transcription factor, I want to find a binding motif, conserve the binding motif. And then when you do ChIP-seq and you realize Actually, there's no conservative. That's like just here and still I do not see it, right? So that may be good sign instead of give up. You thinking about that maybe there's a motif has another called the shape 3D, kind of like a commonality between all those DNA binding motif. And indeed, some of the transcription factor has been published in several cell paper that's showing similar. The transcription factor binding is a kind of shape, DNA shape mediated rather than a sequence specific mediated. So again, this is very interesting to us. And so I want to switch to now I told you about the DRM2 story. And so we want to see another non-CH methylation, which is looking for CMT2 family. And so the reason that CMT2 is so interesting and because it's a plant specific. The human doesn't have to CMT family. And then what is more interesting is even compared to Arabidopsis and the maize, that while Arabidopsis have two versions, 72, 73, maize we have a lump sum called ZMAT2, which is mainly mediating CHG methylation. And so we are very interested in understanding like how does this actual CMT2 mediated CHG methylation, and then how does the two different CMT like recognizing and mediating for different CHG and CHH, and then how do CMT2 even evolve because the animal doesn't have it. And then also the most important is why would the plants even need an additional copy of methyltransferase, right? So really again, kind of starting to addressing this question again, we are collaborating with the GCRACE a couple years ago, and then so these are the crystal structure of CMT2, which is basically the maize version, in complex with DNA, and with similar things that indeed it works very well. And so then what's interesting is, again, we finding these key residues, which is really mediating the DNA and the enzyme interaction. In this case, is RA04 in the maize and R745 in Arabidopsis. And what was also very interesting to us is that when we look in the phylogenetic approaches, and so take a moment to look at the CMT3 is here is always R. Look at that, right? It's always R, but what about CMT2 here? And so we're looking for Arabidopsis, it's V. And then when you go to early evolutionary early plants, it's becoming R. And so yet they actually can show certain level of sequence kind of identity. And so this becoming very interesting. We're thinking that maybe this key V and R is really can determine the sequence specific, methylation specific for these two enzymes. And so obviously the first question is, can we do the swap, right? So it's just a simple experiment because it's just one single amino acid change. And then in CMT2, when you look at the CHG, hemimethyled CHG, it's very minimum activity for the wild type CMT2. But if we change into this V to R, single amino acid substitution, it increased the CHG methylation activity at like a 10 or 10-fold. So this is very exciting. We actually talked to the company people, NEB, and they were so excited that like, okay, now because you can do a lot of manipulating in the vitro, right? So if you do patent and we're going to purchase it because then you have a powerful enzyme to do a lot of things. And so this is another thing we're using another way to do the in vivo, which is again using complementation, looking for bisulfite sequencing. And so this is looking for the CHG, which is blue. And so in Colombia, you have higher when you're in the CC with basically 72, 73 double mutant, you loss of the CHG. And now if you're complementing the wild type 72, it does not come back. But if you have a VR mutation of 72, the CHG methylation comes back. So this is reinforcing the idea that this single amino acid really mediating the difference preference. And so now then we want to see how it actually evolved, right? And so we all dealing with the plant is called what? Duplication. Every single people I talk, it's like, oh, my gene has a different version. When I have to double mutant, triple mutant, right? Same thing. Why? Because in the angiosperm stage, we have the whole plant called the whole genome duplication. So meaning that every single kind of copy of the gene gets duplicated. And then the destiny of each gene has a different way. Some of them like becoming loss of function, they are not functional, so only one key copy. Some becoming very functional, becoming redundant. You have to make the double mutants. And in this case, we're thinking it's a new function. So the idea we have is early evolution in the CMT2 is that you have ancestral CMT3. When before the genome duplication, you have just one copy of CMT3. Right? And then during the genome copy, you have two copies. Now you have copy 173, copy 273. And then somehow, because we have some data showing that, that originally 73, one of the copy just maintained the CHG methylation activity. But then the new one that becoming 72 because have repeat elements. And then the early version 72 still recognizes CHG methylation. Interestingly, quite interestingly. And but then after like over the kind of course of evolution, then these CMT2 somehow diverge to kind of like make more CHG methylation, CHH methylation. And so now that's how we look at the Arabidopsis of today is you have two copies, one of them for CHG, one of them for CHH. And so this is quite interesting. We are very excited because the paper just got accepted last week. And so, we are thinking about them, what is the function, right? So why you need to have a newer function? So CHH obviously has already protein to work on it. Why would the CMT2 have to evolve the new function? And so we're thinking that the two hypotheses that we're thinking about, the first is we're thinking that because the emergence of them decompensate the loss of this called the very old fashion of DNMT3. And so the idea is that again, when we're looking all the phylogenetic trees, and then so DNMT2 is everywhere, CMT3 everywhere. But look at the DNMT3, the only existing in this old kind of ancient plants. And then the emergence of CMT2 is correlated with the loss of the DNMT3. So suggesting that maybe when you have loss and you have the new things coming and to compensate the loss. And so to support this idea that there's one paper just published using this mouse DNMT, and then if you lost this DNMT, that actually loss of CHH methylation rather than CHG methylation. So this is suggesting that indeed maybe the emergence of 72 is to compensate the loss of DNMT3-mediated CHH methylation. But I think we have another hypothesis with you. We think it's more kind of like important and interesting is we're thinking that the CMT2 is also involved to make the plants more adaptive and plastic to the environment. So how do we get in this hypothesis and the conclusion is, again, we took the advantage of this 1001 epigenome and the genome project and looking for the existing 72 mutation. And we are surprisingly finding that unlike the DRM2 and 73 and MET1, they have literally no mutation. And then somehow the 72 is very tolerant to all type of mutation. And so they are existing in all of the places in the natural population. And when we look at where the mutation occurs, of course, the mutation are all, like if you have there's all the mutation at the N terminal, which is newly involved and distinct from the 73. And then, so this is the idea that we're thinking about the N terminal is really a kind of thermal sensor or environmental sensor that mediating the plasticity. So at this stage, we are currently investigating that hypothesis. But what makes things that more interesting now, we don't stop here, and we want to know what's going on in the natural population. And so we are very lucky to really kind of collaborate with Professor Zhong from Fudan University, who is an ecologist. And so they happen to have isolating like some Arabidopsis accession from the Tibet, which is we call it the loop of the world. And then, so when we took those kind of Arabidopsis back to the laboratory condition and go side by side, and so we named this called the Lhasa because that's the city that is Tibet famous for, and that's very close. So this nice thing is that you can just name whatever you isolating, and we can like Columbia is actually named because of the Missouri Columbia, right? Hopefully, I don't know what do we have a Purdue accession? Maybe, right? So that will be a good undergraduate class. Go out to the version, right? And so, yeah, when you kind of do side by side of this laboratory group Columbia versus the Lhasa 2, and then they are quite different. They look different because they are the wild plants that have been growing outside for the whole life cycle, right? And so when we took them went back and then do the whole genome sequencing and also whole genome bisulfite sequencing for DNA methylation, what we're seeing here is Lhasa, which is using this blue one, and the wild type is in the black one. This is the 5' chromosome, and in the heterochromatin region, you see the higher DNA methylation, in particular of CHH for the wild type, but then got us kind of like reduced in the Lhasa. And so we were thinking about and then we becoming very interesting to see whether this plant has a memory, because then we took the plants from the wild and then let them to grow in the laboratory condition for many generations. In this case, we call it G0, which basically just seeds from the wild, and then G1, G2, G3. And we actually happen to have two plant population came from the same regions. And what was interesting is that looking for using the flowering time as a way to testing the memory. So when you look at the Lhasa kind of one, so it's just maintaining the same kind of late flowering even after 3 generations, transferring back to the laboratory conditions. And interesting that we have this Lhasa 2, which is actually mutation in 72, right? And so we finding that now they lost the memory after 3 generations. I'm pretty sure if you starting with the 4th generation, it will be completely Columbia wild type. And so this is quite interesting that we are currently investigating, how the 72 maintain this memory. And so looking for the morphological, physiological, and the stress response. And we also really looking for the epigenetic memory and in terms of transgenerational inheritance and also the adaptation. So now we can using this kind of material to really find a way to engineer plants that can be surviving. Because those plants are not only like flowering, if you look at all type of stress, they are very tough plants. Actually, I did not show the data is these plants are like a heat tolerance, like the UV tolerance, because they're tough, But I mean, at least in the 3 generations. So we are really trying using this as an opportunity to learn something new and help us to engineering tough plants that handling a kind of complex array of environmental stress. Okay, so I just tell you about some, individual level. And so we are also really interested in looking for the DNA methylation in the natural population level. And so this is really driven by our talented postdoc, Jay Liu. And now he's also a step his own lab and bigger than mine. So again, that this is one thing that I feel so proud that, when my people came over there and they are so successful, I'm more than happy than my own success. So it's really, really a kind of like a privilege to working with an outstanding group of people in the lab. And so what Jay came, because he has a background of GWAS, like using this kind of genome-wide association kind of scale. And so when he comes to the lab, that our postdoc came to the lab has diverse background. I don't need them to be expert on epigenetics, but they are expert on their own things. And so when Jay is coming, he is very interested by using GWAS populational approach to identify genetic basis of certain traits. And so when he came to the lab, let's do EWAS, right? So meaning that you're using the epigenetic mark as a trait and looking for genetic basis. And so in this case, he did all this EWAS analysis and using DNA methylation level as a trait, and particularly focusing on non-coding RNA, because we love talk about a lot of genetic has already done, non-coding RNA hasn't been done yet. And so here is showing a complicated kind of diagram, but just simplify that you are looking at the 3 layers of things, right? So the inner layer looking for CG, and then middle layer CHG, and outer layer CHH, and then the different color representing this called CV, which is called efficient variation, meaning that the orange has bigger variation versus the green has a very minimum variation. And so as you can see, a quick take-home messaging from this, you don't need to understand, you just see that there's a lot of variation, methylation in all type of non-coding RNA. And so then we can look at the individual cases And so this is again very complicated than MetaBlot kit map. And so looking for this, we are looking for y-axis is each line of the the axis is individual microRNA, okay? And then each of these kind of horizontal line is X section. So basically what you see here is that like when you have a same color, meaning that the individual microRNA have the same type of expression along all the X section. And so what was different here is looking at, you see that this one RNA has a higher kind of expression all over the places except in those X sections. So you see actually variation among population. And so using this kind of way, and then we find that we actually identify this method QTL. Basically, it's very much like a QTL, but now we're using this called methylation QTL, called MQTL. And we're finding what is the commonality between MQTL. Why we do this? Do we see this bigger methylation violation? And what is their commonality? And what was interesting, we finding that the one key kind of feature came out with the commonality of this MQTL is called plant adaptation traits. And so the first hit is actually fitness, and then flowering, and then plant structural, and then adaptation. So this is quite interesting, suggesting this kind of like a global methylation violation is really a way to tackling the kind of adaptation for stress environment. And so then we look at the GWAS Manhattan kind of like a plot. And of those of you who are not familiar with Manhattan, don't worry about, you're just looking, so this is just an approach to identify a genes that are responsible for different traits. And so when you look at this, you look at the CG, look at there's a gene in chromosome 1, and then finding the same chromosome 1 genes actually also regulating CHH. And so we want to know what is that exactly the gene in the chromosome 1. And then when you kind of like zoom in, you're finding that actually one gene that is called microRNA 157A, which is quite interesting to us because it's a non-coding RNA, microRNA regulating the gene expression. And so when we try to study the function of microRNA, we're using two ways. One is knockdown, one is overexpression. And so we, for those of you who are not working on microRNA, you cannot actually knock out, because once you knock out, you have no function because you have a compensation mechanism. So what you can work on is knockdown. So when you look at the knockdown, we actually see the reduction of this microRNA, and then when you overexpression, of course, you see it, and then you actually see the similar phenotype, like the knockdown showing early flower, overexpression showing the late flower. And so next, we want to know, now we see the correlation, right? We're finding identification microRNA methylation status that really contributing flowering. And so really we want to know the causation, right? So all the genomic, all the GWAS are really correlative and cause switch. So we want to know that how can we only manipulating methylation of one genes, and then what happens? And so if this is a really contributing phenotype, then we only manipulating the methylation state, and it should be recapture the phenotype. And so we are using this called epigenome engineering approaches. Basically, that we're using this dCas9 systems that are using the Cas9, but the guide RNA are kind of like a teasering this DNA method transfers and using guide RNA to go to that specific regions of the genome. Right, so the idea is we are not touching everything else in the genome and only that place to do the DNA methylation. We want to methyl transferase go that place to methylating, like methylating only you, but not everybody else. And so then this is how it works out. We are trying to only methylating this microRNA 157. And when you look at the DNA methylation, indeed, we call this Suntec MQ1 that indeed is showing a higher DNA methylation. And then when you have a higher DNA methylation, then you have a reduced transcription of precursor, you have a reduced of microRNA. And when you have a reduced microRNA, you have early flower. And these are all like microRNA targeted gene. They call it SBL. When you have a reduced microRNA, you have over accumulating of a targeted gene. And so this is really suggesting that, indeed our kind of like a GWAS approach really is a new way to identify a new player in the sense of populational level. And so, and then we finding a lot of MQPL really kind of correlate with the plant adaptation. And so we are using this called the epigenome editing approaches is really a way to providing a direct evidence for the causation of DNA methylation on the non-coding RNA gene by local specific genome editing or epigenome editing approach. I'm going to switch to the gear to, we talk about a lot of DNA methylation. And so I'm very, as I kind of introduced earlier, we're also very interested in the histone modification. And so one thing that we came out very interesting is called this EBS protein, which is really quite kind of fascinating because EBS is tiny, but then yet it's multifunctional. So the EBS has this VH domain that actually binds K27 trimethylation and mediated silencing and maintain the plants to be a vegetative growth. And so if you, and then PhD, EBS also has another domain called PhD domain that recognizing H3K4 trimethylation. And then once you have that kind of activation, then you activate the gene expression, and then the plants transition from vegetative to the reproduction, in this case, flowering. And so this is a highly balanced approach because it's very dynamic. And so now how do you know plants are ready for flower, not ready for flower? Because we have a signaling pathways. And so you know that, like if you talk about the epigenetic people, the two words you keep saying is activation, silencing, right? And so this is apparent contradicting interest of silence and activation is really, we're thinking about is a kind of dogfight, right? And so we are very interested in is like, it has to be kind of balanced. There's no, like life is never black and white. It's always gray. It always have compromised something. And so we are very interested in, you know, is a way of balancing both. And so we are finding this because when we started the lab almost 10 years ago, and so we are very interested in kind of, you know, screen that called readers. And so we talk about kids using this epigenetic code, which basically very much like a genetic code. You have one key, one gene, like recognizing. So epigenetic, you have different modification. You have specific kind of protein recognizing this modification mark and then translating modification mark into transcriptional and chromatin landscape. And so this idea is we are trying to using this called histone peptide array. So for example, if any of you are working on a given protein and you thinking that actually this could be a bind to the chromatin marks, here we go, we can using the same method, right? So you basically, these are like very much like a kind of slides. So that contains all type of histone modification on the slides. And so if you have your protein A, CTR1, right? And thinking that, okay, I'm just recombinantly purify my CTR1 genes and then to incubating on the slides and then like Western blot wash and the antibody to recognize it. So again, that's a typical imaging that you're looking for. That again, the green is just a peptides that showing the all type of modification on the slides. And then the red is you labeling your red with antibody and then protein of your interest. And when it's orange is what? The overlap between the green and the red. So meaning that your particular protein binds to specific modification, and that's how the overlap comes from. And so using these systems that we start to screen, you can screen on everything that you are interested in, right? And so we screen a lot, and then one thing that came out with that screen, which is fascinating, and we decided to follow up, is this called EBS. It's early both in short days. And so when you look at this protein, it's only very, very tiny. And so why it's so interesting? Because we finding it actually this protein binds to K4 and K27. And all those people we know K4 is activation, K27 is silencing. And so that's becoming very interesting. Why would one protein bind to both activation and silencing mark, right? And so like before we getting too excited, we have to convince ourselves we need a multiple evidence, right? So we did a lot of evidence. We're doing like a called peptide proton assay. We indeed finding this protein actually binds both K4 and K27. And we also did the called this ITC. For those of you who are not really or familiar with this, the lower the number, the higher affinity. So basically that K27 is indeed a strong binder for the EBS. And so then we deciding to see what's going on. Again, I'm very, very fortunate to have all people who are crystal structure people who are very interested of everything we're working on. And so when I talked to my collaborator, Jamu, and then he said, oh, that's crystallized. All right. So that, after like several months of try, and so they actually got structure, which is quite exciting because, when you look at that, we're showing that actually indeed, structure of EBS is showing that the actual H3K27 was pi by BH domain. And back to that time, that is already a very big discovery because the BH and then K27 reader is not a very, very common one to be identified. And yet we actually find it another interesting because we know that it's also binding K4, right? And so we have multiple evidence already showing here, but however, regardless of how many efforts we tried, we just could not get a structure with the K4. And so we have very, very kind of day and night discussion because that's like, that doesn't make sense, right? And then we look at finally very closely, we find this called auto-inhibition loop. And we finding that this C terminal, which is shown in blue here, that somehow the auto-inhibiting blocking the PhD domain, which is showing the purple. And so we're thinking that maybe this is why because it's auto-inhibition, it's blocking. And so if we remove this inhibition loop, then it should be able to expose the PhD domain and then should be able to bind to H3K4. And so that's exactly actually the case. When we actually make a deletion mutant without this C terminal, now the K4 is happily and then kind of very strongly binds to the PhD domain. And so this is quite interesting, exciting that because a lot of time, like not every hypothesis always work, right? And this is one of this rewarding moment. You have a hypothesis and it works, right? And so I think this is quite interesting. And then what is more exciting to us is we actually finding so-called epigenetic switch, because we measure this binding affinity by this ITC experiments. And when you look at it for the full length, the K27 K4 trimethylation is this high. This high meaning it's lower binding, right? But when you're deleting this C terminal, that actually the binding affinity improve about 2 to 3 folds. And so this is quite exciting because again, we have a lot of biological in vivo ChIP-seq and flowering things that all showing they actually binding to both K4 and the K27, and they are regulating epigenetic switch. And so, but to kind of really sum up all those things, and so here is what we're thinking about, and our model is that this EBS protein has a BH domain and a PhD domain. And so in the vegetative growth state condition, so the BH binds to the K27 char and then recruiting the PRC complex and spreading this silencing map and maintaining this chromatin state to be more close. And therefore the key genes like FT, which is a flower gene, start to be silenced. And somehow that during floral transition, it could be flowering, it could be sick development sick, it could be environmental sick, or hormone sickness, and tell plants, it's ready, we should flowering. And so we're thinking that maybe this somehow that this auto-inhibition start to be kind of like the release out. And now this releasing of auto-inhibition allow PhD domain to bind H3K4 trimethylation and then preventing kick out the PRC complex and now allow the chromatin start to be more relaxed, express FT, and plants are ready for transition to the reproductive stage. And so we are really kind of working on this kind of model, and we have given the idea that we're thinking also, like in addition to that, we are working on the biomolecular condensates, and so we are going to have more kind of like stuff coming out from this type of studies. All right, I think I have last 10 minutes, so I'm going to tell you about the new toys that we're thinking is pretty cool that I want to share with you guys. And so, we talk about that. Come back to DNA methylation. I give you a different flavor of histone modification. Now we have come back to DNA methylation. So, like for people who are not really essentially at the core of epigenetics, you will always think DNA methylation always associated with silencing. It's not necessarily true. So, if you're thinking about methylation is a sequence context, position context that matters, and that's how it works out, is that when you have a transposon silencing, yeah, everywhere it's silencing. But if you're methylation in promoter, then you always associate gene silencing. But there's a tons of the studies showing that actually the gene body methylation, main DNA methylation in the body of the genes, and these genes tends to be like medially expressed gene. So not necessarily associated with transcriptional silencing. And so this is like a quite interesting for like where what is the mechanism, what is targeting, what is the like the maintenance and inheritance. And then we realized that all the like that was literally when I set up a lab, and how can we actually able to tackling this question? Because all the methyltransfers, if we mutate, are they all going to knock out everything, right? And so we are thinking about, can we developing a new systems that doing engineering and then very clean system with that. And so so this is again another talented postdoc who just also established his own lab, is developing this called cis engineering of local specific kind of like a knock-in systems. And so when why the knock-in system has been well established probably in the mammals, the plant are very limited step. And so we decided, okay, let's try. So how the knock-in system working out is basically you knock in a piece of DNA into the place that you want to be in. Simple, straightforward, right? People now what we're doing, we are doing transgenes. Like we're doing transgene, we're taking a transgene of RTCCP1 genes, right? And then we usually say, oh, we need to even knock out the background because we want to only the function of its transgene in the endogenous promoter. So if we have the knock-in system, let's not worry about all this problem. So basically, all you does is you can make a tag line in the original place of the genes, and they are expressing its own promoter in the same location, and then you have a tracing system. And so this is ideal, but using this idea, we are very interested in can we utilize this opportunity to engineering a repeat elements. A so-called repeat elements is kind of helpful converting to really recapture these already very early events, like how the hyperchromy start, initiate, maintained, spreading, and then inheritance. And so we're thinking that because the current established using genetic mutants of the DNA methylation mutants, they already established as level. And when you go through like a lot of genetics, there's no way you can trace in the dynamics. And so this is really the motivation. And so how we do that? And so basically we are using original, we borrow a Jiankang juice kind of like a systems. And then originally, which has a one-time system that basically you are using the Cas9, dCas9 that using called XL specific promoter, so that this editing or knocking only occurs in the germ cells. And so that will be kind of transmitted to the next generation. You don't need to do a lot of kind of genome typing. And then you need to have a kind of donor DNA that because donor DNA meaning you integrating your DNA piece in the genome. And so you have a guide RNA. The donor DNA and guide RNA are in the same vector. And so in this case, that we're using this ABI5 as a way of just a report a line. So it can be anything, but the way that because we establish a system, we have the few things meeting criteria is it has to be very easy for screening. And so we don't need to have a lot of time. So because the ABI5, why we're choosing ABI5 is in the wild type is sensitive to the ABA treatment. And if in the mutated version, it's insensitive, and therefore you actually see a lot of regular growing. And so this kind of mutant phenotype will tell us that, like when you inserted the repeat elements, how will trigger the silencing. And so that when we inserted this repeat elements, one way we're using this called the MTRBC because we are only dealing with one locus. We don't need to do whole genome by sequencing. And so how MSRBC working is basically that if you're looking for so if you see, because it's sensitive, insensitive to the DNA methylation. So if you have a methylation, then it will be cardiac. And so when you do amplification, you do not see it. So basically that when you see this kind of reduced the bands, suggesting you have a higher DNA methylation. And back to that time, we have no idea like how many repeated copy is sufficient. And so we just did one copy, 2 copy, 5 copy, 13 copy. It's just like a random things. And we finding that actually 2 copy is sufficient. When you have 1 copy, you still see the methylation. You start to see the more methylation. And the same thing that when we look at the different copy and we finding that looking for H3K9, and we see the H3K9 is really also start to kind of like accumulating in this insertion site. And then, so then we want to kind of like a using system to ask when you have the insertion of the repeat, does it affect the transcription, right? And so again, because that this is a really called a way of very easy for us to look in the phenotype. Because if you have a regular transcription, then it will be sensitive to the ABA, right? But if you have a blocking of transcription, it will be insensitive. That's just simple, right? Like a mimicking phenotype. But surprisingly, that when we actually inserted the everything, it's like all sensitive, meaning that the transcription somehow just normal. And then so the phenotype and also the RNA also tell us that somehow our repeat elements that start to have no function. And this is another like a nightmare a lot of time, like, oh, you know, you have everything designed and they are not doing something, right? So I tell you an encouraging case that you actually, your hypothesis work beautifully, and this is another case not working. So, you know, what are we doing? Are we stopping? No, we are trying to see what's the reason why the transcription is not blocking. And so we have another, like based on whatever we know, we have another hypothesis. We're thinking that maybe, you know, it's just simply like the methylation stay in the same region is insufficient to blocking transcription. And so maybe you have like a blocking kind of like a pathways. So you have to blocking because methylation at a single nucleotide is not going to function, to my opinion. You have to add kind of critical mass. You have to add kind of blocking the whole way, accepting, and then start to transcriptional silencing. And another hypothesis is maybe, yeah, you actually spreading out, but you have the demethylase as a way of kind of like a kind of like a defense systems, because once you methylation spread into gene, it's not going to be helping gene. And so you have different systems that keep removing. When you're adding methylation, you have a demethylase keep removing. So it's a dogfight. And so while we are still developing this idea that we actually start to quickly testing this blocking factor. And so the reason that we're using this blocking factor is called IBM1, which is a K9 demethylase. And so interesting that when we actually kind of like when we kind of like cross this whole system back into using genetic back to the IBM1 mutant, we actually finding like it becoming insensitive. Did you see that? It's now all green, insensitive. And so when we look at the methylation, now we are using genome by whole genome sequencing because it's just the repeat is so challenging to do like a lot of MCRBC. And so if you look at that if you have wild type IBM1, they are just only strict in this insertion site. But now if you remove the IBM1, then they start to spreading out. And so this is the same thing for the K9. And then so this is really like we're thinking now the IBM1, now we want to know whether it's inherited. And so again, to answer that question, we take the advantage of genetics. And so we basically we're using backcross. So you backcross that, you know, getting a normal IBM1, getting a normal kind of like genes. And so if it's inherited, then it should be insensitive. And if it's kind of not inherited, it should be sensitive. So that's called inheritance. And so we actually successful, this is showing F3, we're actually showing F5 generation. And we can see some of the line actually showing kind of inheritance. And so same thing that we actually looking for DNA methylation and transcription, and we are also in its methylation. So and this is my last slide that we are just quickly to tell you how using this called the knock-in epigenetic knock-in systems that really creating a new way of epi allele. And so the same path that can be utilized by any of you guys who are interested in using this type of systems is basically here we are trying to knock in a repeat elements, but you can knock in like a PRC element, let's say, right? You are knocking in the some of the microRNA target genes, and then you are knocking the tag line, right? And so basically what we're finding that when we initially kind of knock in the repeat elements, it's nucleation, heterochromatin nucleation process involved in a traditional enzyme, which is MET172 and the DRM2. And we're finding that actually the IBM on the histone demethylase is really play role to preventing this demethylation spreading out. And so, but if you remove this methylation, that it can actually really spread out. And so now, now we are removing this original insertion site back to this original DNA sequences, that the methylation marks left stable there, and that's stable for many generations. And if you're thinking about the original material now with the new material, they do not have any DNA sequence change. What is changing is you actually have epigenetic mark behind that, and that can be stable for, in our hands, at least 3 or 5 generations. So, actually, I tell you many stories, and so, I will be really grateful for all the people who are done the excellent work. And then these are current lab people, and that's former people. And I think that I usually acknowledge them during my talk because I will always forget. And then always thanks for all the collaboration, and then all the funding. Without funding, that's not possible with a lot of genome sequencing. So it's very, very money consuming. And thank you very much for your attention, and I will be happy to take any questions.
Are there any students that want to ask any questions first?
Yes, I have one.
Yeah,
So,
great.
thank you so much for your presentation. It was wonderful and so entertaining. So my question is, what you have shown is that there are many persons that goes to your lab, and after that they go to academia and establish their own lab.
Yeah.
So what are the things that you encourage them, and what are the main things that inspire them to continue?
Yeah, that's a great question. So when we try to recruit people, that we are looking for motivated students and motivated people. And then, so we are maintaining, I don't usually have hypotheses. So I would let the discovery driving the project. And so, once you have a discovery, and let's going on, right? And so I think that so another way we try to motivate the people working on multiple projects, so that, that you can ensure like not every project works, right? And the one that I present the story is many work, and then not including a lot of failed experiments, right? So I think that, maintaining curiosity, motivation, and then don't afraid of making a mistake. And even you have a kind of wrong hypothesis, it's okay. Let the data driving your hypothesis, and you're going to discover something. So that will be my kind of 2 cents. Yeah. Yeah.
So one thing I've noticed when you see epigenetic phenotypic variation,
Yeah.
it's always flowering,
I
often
see.
flowering. So is that just because it's easy to see, or is that because that's something that's particularly epigenetic or particularly plastic?
Great question. It's just like I'm really not a plant biologist, and the flower is so obviously to me that there's a lot of kind of variation of epigenetic phenotype, like roots, right?
Mhm.
So I definitely thinking there's a kind of roots, and there's a stem cells, right? And then there's a hormones, and then immunity. It's just me looking for the flower, and then maybe let's see, you talk about not just only me, right? Like most people looking flower, because a lot of people chromatin, we are looking on molecularly. We haven't really start to looking for different biological process. For example, the nitrogen fixation area, right? And then the nodule formation. I'm pretty sure there's a variation. We haven't have enough people to work on different area. And so that's why that my new people, my people coming out my lab, they all working on different, like one people working on soybean, one people on maize, rice, and then the wheat. So they all different, took the training from the lab, and then working on their own kind of niche in the new biology, that connecting epigenetic with the new phase of biological questions.
So it could be like the guy's looking for his keys under the lamppost because that's where the light is.
Definitely. It's just like it's always easier, and for us, right?
Yeah, we have color in corn. right? Color
Exactly,
kernels.
exactly. So like it's obviously phenotype that you know, you don't want like miss, right? Yeah.
I have a technical question. So you mentioned that the 3D shape of the DNA will determine of the transcription factor binding sites.
Yeah.
So far, is any way to predict the binding site based on the 3D shape?
Yeah, I think there's some program like right now that like the development of AI, right? So there's many computational program you can like feeding the DNA motif, right, sequence. So for example, you get the chip of a particular transcription factor, and you identify like in the traditional using like MEME looking for the binding motif and see enrichment of A, T, C, G, whatever. But now if you finding, oh, I pull down my DNA and then cannot find any motif. So you can take all the DNA pieces that you identify from chip experiment and feed it into the program, and they will kind of making the prediction.
Okay, so we have to have chip seq first and then.
Alternatively, if you have a hypothesis, you're thinking that these are the key genes that could be potentially regulating by my transcription factor, then you can take the genomic and take that piece of DNA doing the simulation, right? So that's two ways, theory or computational or like a experiment-based evidence. Like many of you guys maybe have RNA-seq data that in the knockout of transcription factor, I see 1,000, the genes upregulated, right? And so you may take that as an opportunity. So I want to take a promoter region of this 1,000 gene and feed into my model and see whether there's any commonality come up. Yeah, so just like using a lot of computational skill that can maybe help you to develop new ideas. So that's all I'm saying, that a lot of time we go to one end and then we look back and like, oh, maybe that's go another way. Okay, go another way. So that's how it works out. Not every hypothesis works out.
Okay,
I think that we have one more. Yeah.
Okay, thanks. So I was wondering with your accession from Tibet, you showed in one of the slides that it had lower CHF methylation.
Yeah.
And a later slide, you showed there are two populations. One of them forgets and the other remembers.
Yeah.
Which one was the one that had lower CHF methylation?
So in the methylation for the LASA2, which has a lower DNA methylation and then forget.
And that's directly into the CMP2 mutation.
Yeah, so and that's got 72 mutants. And so we're thinking that 72 maybe a forgetful mix. So well, it's easy to say that.
Okay, I ask one more question and then.
We actually have one more student asking.
We have, it's actually a follow-up.
Okay.
Is that kind of lost memory process, could it be reversible? Like, when you bring it back?
You bring back to the 72?
Yeah.
Yeah, that's possible. Yeah, so you can, yeah. So now, right now we're doing the log generation, right? So we can also try to put back and see whether or not they gain as memory. So that's possible. Do you have an urgent question?
Yeah.
For us? Taking the advantage of the.
That's my question.
Uh-huh.
So I was wondering about this small tiny EBS protein.
Yeah.
We have an autoinhibitor domain. I just wondering how they regulate it. So most of the time, like if the protein is kinase, the autophosphorylation is
Yeah,
kind of
yeah.
key to
We have 3 people working on this.
releasing this inhibitor domain, but this protein is not kinase.
Yeah,
So I'm
yeah.
just wondering, is there any other modification or any attractive protein?
Yeah.
This mechanism?
Yeah. Again, we are writing 3 big paper now. And so I think one of the idea is actually, it's a kinase, irregular by kinase, and then so form a condensation, molecular condensation response to stress signals and nutrients. And then, so we are connecting a lot of dots. And we are also trying to, because the C-terminal auto inhibition has the simulation, ubiquitination, phosphorylation, and we are identifying many kind of residues and making like, that's what I was really intrigued about all your kind of like protoplast work, because we also see degradation, protein degradation, ubiquitination, and then kinase. So indeed, we actually identify kinase and we identify key residues that kinase regulating. So there's a lot of things going on we can Yeah, hopefully next year, if you guys invite me again, I'll share. But thank you so much.